all, I really want to thank the organizers for inviting me back to ESI. It's basically my first visit back since I left, more or less. And it's super exciting to be here, to meet familiar faces, and also to be part of this really exciting conference. I also want to thank the organizers for an impeccable organization. The conference has been really well organized and everything has been working so good. And I've been also given so much liberties when I was invited to talk about so many different aspects of my research. So Matthias, when you sent that initial email, you gave me 3 different avenues of research that I do that I could talk about. That was made me very, very excited. So excited that I decided to also bring along 3 different students with me, Nir, Lorenzo, and Gal, who have actually described quite a lot about the process-based approach to the study of time perception and other things in their really interesting posters that are still hanging outside. So with their presence here as my kind of fortifying team, I do encourage you to go to their posters if you really want to hear about the work we do about time perception. I have, however, changed the title of the talk and also changed the topic of the talk to talk about really work that is entirely new, will be submitted very soon. And I think it's work that is relevant to anyone interested in timing, but also to anyone interested in understanding neural mechanisms that we could learn about from looking at spectral data of fields. So, and this is actually looping into another circle that is closing here at ESI. Golan Kravat was also a PhD here at ESI. He worked with Ilka Dister with rats and somatosensory cortex. And then after his PhD, he was trying to figure out what the next thing will be. He decided to start doing cognitive neuroscience and approached me asking me, hi, I would like to do cognitive neuroscience. Could you teach me to do that as a postdoc? And I, of course, was very happy to close another little Frankfurt loop. So Golan visited and stayed in my lab for a period of time. And in that period of time, I put him on some timing tasks. But as you will see, we took it to slightly different directions. So the title of the talk is Universal Rhythmic Architecture Uncovers Distinct Modes of Neural Dynamics. I will reveal to all of you that I am stepping outside my comfort zone. Most of my talks really deal with cognition and how neural mechanisms and biological mechanisms explain behavior. I think it's all very highly relevant to this talk, but this talk really is a bit more nerdy than most of my talks. So let's just kind of get a guide to what this talk is going to be, or the Hitchhiker's Guide to the Universal Rhythmic Architecture. I will start with a historical background, understanding how we got here, and it is really related to looking at rhythmicity in time perception. So the first task that Golan had to kind of be tasked with when he joined the lab. But as you will see, our very initial findings very readily will make us take a slight detour to talk about oscillatory bands, about the state of affairs, and about some serious challenges that we have when we talk about rhythmic phenomena and oscillatory bands in brain activity. I will, in this process, introduce to you a measure for rhythmicity, which uses some smart measures that were already out there, but kind of derives them to do something beyond what they did at that point, and will claim that these rhythmicity measures help us uncover a universal architecture in the spectrum. We will then try to better understand the single trial manifestation of rhythmicity in this context. And finally, we will hypothesize or entertain some possibilities on whether this rhythmicity can uncover neural modes of neural dynamics. So let's start with time perception. I already said that Nir has a really interesting poster out there, but Nir, in his PhD, collected more data. He also collected data on this quite classical task in which people have to segregate 2 visual flashes. The contrast on this projector is not great, so I will actually try and see what you see in order to help you understand what you're seeing. So in this task, we generally presented a grating. I mean, I come from Deutsche Ordnung 46, so of course I'm presenting a grating on the screen. And after that grating, think of it as context, high contrast context to the systems, high context drive. There are these white flashes. So this here is a white flash as well as this, okay? And in this case, there's a little gap ranging 7 to 56 milliseconds between these white flashes, but sometimes there was no gap, okay? And the subjects have to decide, was it one flash or were there two flashes? And what we found, very robustly, is that if we change the contrast of the context, the thresholds change dramatically. So there's a shorter threshold when the grating is high contrast. And this is published work with some interesting ideas that we already put out there. And this is, I'll just draw your attention to the psychophysics. This laser pointer is very sophisticated, but a little bit lagging in time, so apologies. And what you can see here is the case of 100% contrast, 50% contrast, and then when there's no annulus at all. And as you can see, it's, when you have a high contrast context, you're much better at detecting that gap. You can detect it when it is much shorter. That was the behavioral finding. Of course, the next thing we wanted to do is understand what are the neural mechanisms or what are the neural manifestations that could help us understand why that happens. So we had EEG measurements from a full scalp of electrodes, and looking at 3 electrodes from the back of the head, as one does. I'm showing you subjects' data. This is a single subject evoked related potential. And you can see 2 cases here. This is locked to the grating onset. And what you could see is that the grating obviously evokes this kind of ERP, this stereotypical response that a lot of cognitive neuroscience spends time describing. And after that, there is this kind of interesting fluctuation. I was quite surprised. Typically, when we do time averaging, we don't see this oscillation so strong. But we saw it in this particular subject, so we got kind of excited. And we wanted to characterize it in the spectral domain. So we wanted to see what is the properties of frequency of this. And the typical way to go about that is to look at some measure of power. So we use nice state-of-the-art measures for power. We're using this Raoul's supralets here, another Frankfurt alum who is contributing interesting methods to the field. And what I'm showing here is just frequency over time for the high contrast, for the low contrast. And if something is encompassed in a black contour, it means that's a significant cluster. So based on work that I've also looked at before, we see a high frequency increase in power over the baseline, which is kind of accounted for here. We also see in this group data this kind of big blob, likely reflecting this nice ERP that we also anticipate to see. However, this interesting clearly eyeballed phenomenon is not obviously there, and it's definitely not significant. So that made us kind of think, hey, that's kind of strange. Like typically it's the opposite. Typically you see nothing in your time average data, and then if you use the right kind of spectral decomposition, you might pull something out of the data. So in order to move forward, we turned to another property that we have in oscillations. We wanted to turn to a measure that will be focusing on the phase. So the phase, I probably don't need to explain too much here, but the phase of a signal would be kind of where are we at in the cycle? Are we at the peak? Are we at the trough? And in rhythmicity, generally, when we see this type of dynamic, we assume that a rhythmic phenomenon will be replicating itself. It will repeat over time. So this cycle will be good at producing a prediction for the next cycle if something is truly fluctuating. We turn to a nice paper from Anne Franzen with Freek van Ede and Eric Maris, who have quantified a similar quantity and termed it lagged coherence. And the idea is it measures rhythmicity, which is defined as the consistency of the phase relationship between time points that are separated by some interval. Let's take an intuitive look at what that means. So if I filter my data at 10 hertz in this case, I could take my signal, and then I could take a displaced version of the signal. So it's the same signal moved by a certain lag. And I could go point by point and ask what is the phase difference between these two signals. And I do that for all points. And generally speaking, if all the arrows point to the same direction, it means that there is phase consistency over time. And even when they're transitions, they're kind of smoothly progressing. So we could plot this on a polar plot. which results in this kind of display. And then the lagged coherence would basically be the vector sum of all these phase differences, okay? So this would be a lagged coherence of 0.63. So lagged coherence, that would be a decent lagged coherence. And these researchers were mostly interested in looking at these phenomena and asking, how long does this last, okay? So they used to look at these type of plots in which they will kind of compute lagged coherence over different cycles for a given frequency and plot the lagged coherence. So there is some kind of decay, obviously. Another illustration now from 16 hertz, where we see something different. Let's just look at the data. This time course seems to have moments which are a little bit periodic, but then there seems to be kind of phase glitches a little bit. And if we take that signal and look at the phase differences to itself after a lag shift, we will see that the phases are really dispersed all around the polar plot, resulting in a smaller vector sum, a smaller lagged coherence. So now we could replace this figure and talk about how do these phenomena extend in time for different frequencies. This is nice, but it still wouldn't help us recover the phenomenon that we were looking at here, right? Because this is basically looking at the entire signal and really kind of thinking about how the different lags are affecting, how is the displacement over time affecting the phenomenon. So we took the lag coherence and we kind of tweaked it a little bit in order to be able to say something about a time-resolved rhythmicity measure. So we call it the within-trial phase lock. And if we take this perfect sine, we are looking at a given time point. What we can do is very similar to what Eric Maris and his team did. We could look at one cycle at this frequency ahead, so one cycle in time at a given frequency, and ask, what is the phase difference between these 2 points of the signal? We could do the same for this t0, but this time going backwards in time, and ask, what is the phase difference? Now, if the signal is so beautifully periodic, then obviously the vector summation of these 2 values will be quite large. And if it's a perfect sine wave, we will have a WTPL of 1. If we introduce some kind of phase glitch into this signal, then obviously the phases of these differences will be very different, resulting in a smaller or close to 0 WTPL. And this is basically a single trial measure. You could look at it at a given time point, and you could then move time point by time point and see, do you have rhythmicity? Where do you have it in frequency, as well as when in time? So we could do a lot of things with this. We could go back to our signal. We could describe this particular subject and find that indeed there is a very strong rhythmicity going on where we thought we saw it. We could also see other phenomena that might have not been eyeballed. We could do statistics at the individual subject level because we are quantifying this at a single trial level. And we can also, of course, do group statistics and see these type of effects. So that was just kind of a short methodological interlude, which we will get back to. But I wanted to just kind of, again, look at the data for a moment and share an observation. We were looking at these data. I didn't say it before, but this picture actually is baseline. So it's important to baseline because obviously, for obvious reasons, but even when we don't baseline, which is this figure, we seem to see a very, very robust dynamic with these kind of different type of phenomena very clearly in the WTPL. And when we looked at this, we were kind of thinking, hey, wait, hang on, maybe these are the frequency bands. Maybe this method is very, very good at actually kind of pulling out this notion of frequency band, which I will talk about in a second. It kind of, stuff we've read about, but sometimes struggled, particularly when answering reviewer 2 and such. Another thing that we've noticed is that when we have these kind of increased rhythmicity moments, in between, often we have these moments of decreased rhythmicity. So the first thing we thought is, hey, maybe those are the borders. Maybe we actually managed to kind of empirically measure borders between bands. That would be very, very interesting. Or maybe we're talking about a completely different phenomenon, physiological phenomenon, which we should interrogate. So this was enticing. And with this, I want to now just kind of switch gears a little bit and talk a moment about oscillatory bands. Again, this crowd is quite versed in this, but I want to kind of just share with you what I see as an assumption in this field and challenge it from several different angles. So we've all kind of read about canonical bands and frequencies and oscillations, and we've all were given this kind of consensual papers where people really talk about what is the delta, the theta, the alpha, the beta, the gamma band. People know that using words is often kind of misguiding, but people also know that having words helps us talk to each other. And generally speaking, and this is just one kind of example from Bujaki, there is this idea in neuroscience that if you look at the data in the frequency domain, you can see these frequency bands. This here is in a logarithmic scale, and you could see them linearly kind of covering the entire range of frequencies in a way that is continuously, seamlessly kind of transitioning from one band to the other. Now, this assumption, I think is challenged on at least 4 different grounds that I will talk about now. First of all, there's the definition variability, so taxonomy, right? I said that we need to talk to each other. I said that we gave, names to phenomena. Some of these phenomena were named in this building, actually, which makes this kind of the talk of my choice for today. But when you actually try and survey the literature on how people define the borders between the bands, it is kind of messy and variable. There's a lot of variability in the definitions. And you could take a moment to look at this figure, but we have a lot to cover, so I'll go right to the next challenge. It's not, I don't have a good title for it. You could think of it as the gap between the bands. So I'm showing you a picture now from what is considered more or less state-of-the-art in parameterizing of spectrum. This is a figure kind of from the Foof paper from Donahue. And you could think of it as what are the gaps between the bands. You could also think about it from a perspective of what is the baseline. We know that spectra have this 1 over f feature. There is this exponential feature in the data that is always there. And what Foof has brought to our field is the ability to remove that trend in order to better characterize these 2 bumps, for example. And these 2 bumps are basically power measure, and they can help us kind of separate a little bit of the bumpiness from some kind of noise. But first of all, the noise is not well defined. And you could think of it as what are the gaps between the bands? What do we know about these regions in terms of the physiology that is characterized there? The other problem relates to statistics, actually, right? It's not so clear how we could say that something is statistically significant or statistically rhythmic in this measure. Another challenge that we are having to deal with, and I think in beta band this has become very obvious, there's functional distinction. So we have been talking for several decades about beta as one frequency band. But if you just take this recent paper, I'm not going to go into the details of the task, but there are 3 different stimuli, blue, green, and pink. And the animal is sometimes attending to the blue, sometimes to the green, sometimes to the pink. And you can see that if you look at the high beta, which is 21 to 29 defined here, every time the stimulus comes on, there's a drop in this oscillatory frequency. That is not the case for low beta. So low beta cares about one of those stimuli. So if you're attending to the stimulus, when that stimulus comes on, there will be a drop in the activity. So some frequencies are clearly kind of seeking to break off from the taxonomy or from their original band. The last challenge I will talk about relates to physiology. So there is a physiological observation. And this is where, remember Andrea's GIF yesterday of the food fight? So this is, again, this is a food fight. We will not have the food fight here over this. But I will say that, this is what we often imagine when we're talking about rhythmicity, or at least until recently, this is what we have imagined. But increasingly, we see in the literature that when we try to characterize spectral phenomena, they are often not sustained. They're actually very transient. So what are we looking at, and how well can we characterize these different types of phenomena? This is the part where it gets a bit food fighty, and there is a debate in the literature. I think there are debates on like, what are we looking at? Are oscillations sustained, or are they bursty? I will just kind of point to the potential of each type of signal, because I think that the work I will show you today doesn't address the debate, but it could actually provide some tools for the debate or a refined view on how to move forward. Now, when we talk about an ongoing oscillation, this has inspired the idea of phase coding in neural representation. Phase coding is the idea that if we have a state of oscillation, we could start using it in some kind of biological mechanisms. We might want to propose that it could be the substrate of integration in the brain, or we might want to propose some kind of overcoming some scalar displacement in the brain, interareal connectivity. We could kind of build that into the system. We could do a bit more even. We could say, hey, the spikes of the neurons, the outputs, the neuronal output, is actually organized based on that phase. And I think the hippocampus gives us kind of the nice examples of the phase precession, for example, right? So we know that we could actually get added information in neural representation if we take into account not only just firing, but also the sequence in firing. On the other side, you could call it a camp, or you could call it an idea. There's this idea that neuronal representation really is captured in the rate. So if you have a certain population of neurons, a certain state is going to be represented by the firing, the degree of firing, and the time or the temporal relationship between the firing of this population is not so important. And this is a little bit more consistent with this idea that you have some kind of perturbation, say an input to the system. It will create this ripple. We will measure it as a burst. And the burst, hey, it might even be an artifact of our measurement. It might be something we're doing to the signal when there's a phasic input, or maybe it's a biological, local something that is happening. So these are two ideas and a little bit of a color code that we will take over into the talk. We will not address this debate, and I think this has been kind of a debate that we're not touching at the moment. But I think that if we improve how we think of rhythmicity, we might be able to think about this debate in new ways. What will we do? Well, we will challenge the idea that the electrophysiological spectrum is made of seamlessly progressive oscillation bands. And we will do that by looking at our measures of rhythmicity and trying to see whether our measures of rhythmicity might uncover a universal architecture in neural dynamics. So, if there are clarification questions, ask them during the talk. I'll take a sip of water, and then we'll go into the next nerdy part. So remember lagged coherence, and as I said, I think Maris and his team were mostly interested in looking at how these kind of phenomenon, or how a rhythmic phenomenon is prolonged over time. We looked at this figure, and kind of decided to kind of emphasize a different axis, let's say. So if we look just at a single lag, I'm now plotting that slice with frequency on the x-axis and our measure of rhythmicity, which is basically, it is that to measure, but to kind of distinguish it from lag coherence, we're calling it lagged angle vector index. It's going to be called LV, but most of the time I'll talk about rhythmicity. If we just look at the single lag, we see there is a certain morphology to that. And this morphology, we wondered whether it's stable. So we looked at this morphology at several different lags to notice that actually it is quite stable. So now we're doing something that I was actually taught in this building never to do, which is to compare between different frequencies, right? We don't like to do that typically. We like to make sure that we don't do that. Good reasons not to do that sometimes, right? Because of 1/f, et cetera, et cetera. But I am now going to compare different frequencies to each other. And I'm going to say that if I look at this idea of lag coherence in the different lags, there is a shared morphology to this. There are peaks, there are troughs, and it's kind of interesting to look at. If the lag is very, very short, then obviously I'm almost comparing a signal to itself. So I have very high rhythmicity. So I'm kind of at ceiling. There's not much dynamic range. If the lag is super, super long, three cycles here, I'm kind of at floor. Like these are probably phenomena that are very difficult to relate to each other. So there is kind of an optimum of looking at this morphology, which is somewhere kind of around the yellow line, which is kind of at 1.5 lags. And what do we see when we look at that? So I said we see peaks and we see troughs. Another thing that we saw over a lot of data is that the median value of lag coherence in using this lag is around 0.4, okay? So obviously this median will change in different lags, but we picked one case and there's a stable feature over a lot of data, thousands of data sets, where 0.4 is kind of that middle ground. We can call it like our noise level or control, baseline, if you will. There are peaks that rise above this baseline. And they kind of correspond to things we know about the signal. So around 10 hertz, we will call that peak alpha. And maybe we could call the left one theta and the right one beta. These are moments in this spectrum. This is a single subject, and in which there's increased rhythmicity over the baseline. Important to emphasize is that these peaks are detected automatically. So there is an algorithmic way. I mean, basically, we look for the absolutely strongest peak, because that's what thousands of data are showing us, and that will be the alpha. We define that as the alpha, and we don't even need to be very strict. We could say, pick the biggest peak between 5 and 15 hertz, and we will typically find this. And I will go to the population. We'll scale up in a bit. So finally, we have an automated way to identify, in this picture right now, theta, alpha, and beta. And we also have another type of phenomenon, right? We have this baseline at 0.4. We know that we can rise above it, but we can also go below it. And that's kind of interesting and hasn't been quite described. And next, we wanted to know, okay, but should we consider everything that we see here? Like, this seems like a very prominent trough, but what about this? It's quite close to 0.4. So what can we do? We could go back to the data and we could estimate a randomization distribution by basically shuffling phases. So keeping other features of the spectrum at the level of each subject. We take basically their 1/f spectrum, and we then simulate data in order to create indeed a noise ribbon. And now we know that this trough should be considered, but this trough probably not. It's within our noise ribbon. So green is going to be coding things that are above the rhythmicity ribbon, and blue will be denoting those troughs. So that was our single subject, and we have some statistics. But in order to talk about the architecture, or a rhythmic architecture, which is universal, we need to start generalizing and see how robust is this. And the approach that Golan and I took was basically to go to a lot of different data types, and so on and so forth. So let's scale up to the population. This is this subject, automatically detected peak. I could do the same process, and here I'm doing it for 90 subjects, and show the frequency of occurrences of the peak in these 90 subjects. And I get something quite nice and central. I will say that the dashed lines everywhere denote the canonical bands, kind of a choice from the literature. So for alpha, we have a nice distribution. We could, of course, look at other frequencies and basically see that we have kind of nicely organizing automatically detected peaks for the aspects in the spectra that are significantly beyond our noise ribbon. And now I'm starting this type of representation, which we'll repeat. So let's take a moment to understand it. We have the different frequency band names. And we have the frequencies on the x-axis, and each dot now is data from one data set, and the width of this line is the confidence interval, 95% confidence interval. I can do that for the peaks. I can also do that for the troughs. And as you can see, maybe it's not a big deal to show this for alpha, which is it's been around for 100 years, right? But now I'm talking about a novel category in my spectra, and everything, again, is automatically defined, and I see something quite nicely organized. So I can add it now to my taxonomy, let's call it. And basically what this gives me now is kind of novel categories in frequency space, right? So I have a delta theta, I have a theta alpha. My beta apparently has two different type of bands. One which is rhythmic, and one which is a trough. And we'll talk about what exactly is happening in these troughs. The first step towards talking about how general this phenomenon or this architecture is would be to take a look at different data sets. I told you, I think these are Neil's data. These are the data I think it's the original data that I started showing at the beginning of the task, and maybe a couple of other data sets. But I'm now going to add more horizontal elements which come from different data sets collected, some in our lab, some data available online, and some data from labs in Cambridge, which is where Golan is right now. And so some of the data are visual tasks, some it's eyes closed, tactile task, auditory, visual synchronization task, memory, 2 different memory tasks, 178 participants. So this is quite compelling. There's no question that, and another element about this visualization, the percentage basically denotes the proportion of participants, so subject data set, for which we found a peak in that aspect. So as you can see from theta and above, we're doing a pretty good job. So basically all the data that we're looking at seems to have these aspects in their spectrum. So that was EEG. In the next case, we wanted to go bigger, have more data, and this time we're also moving to a different measurement modality. We're looking at MEG, okay, so magnetoencephalography, and we approached Rick Henson, who has the CAMCAN data set. This is a really interesting data set, 625 participants, and what's special about these participants is that these participants range in age from age 18 to 88. And so you can see, first of all, we kind of survived the move from EEG to MEG. It seems quite similar. And we're still doing a very good job in identifying the peaks. So majority of our, like a large majority of our data sets are showing these phenomena, these automatically detected peaks. Another thing that we're seeing is something that to some of you in the crowd won't be a huge surprise, is we're seeing this, this kind of age-related modulation. If there is another laser pointer, I can dance more freely. Where is it? Aha, there we go. Okay, here we go. So what we can also see is that in some frequencies, there seems to be a systematic change in the frequency over age. Now, we've seen this in the literature, and it's nice to see it here. And the star basically denotes that this is a significant modulation in the peak frequency. Okay, now one can ask, okay, so I'm measuring non-invasive physiology. What happens when I go into the cranium? What happens when I start measuring invasively? So the first opportunity we had to measure invasively was brought by the collaboration with Leon Deuel's lab, who is co-mentoring me with Gal Vishne, who is here with us. And this is data that is collected from patients awaiting their surgery for epilepsy. This is an echocorticograph, so the channels are on the cortex, but basically what this brings is the opportunity to generalize to invasive recordings, but not only that, right? It also allows us to ask, okay, do we see this everywhere we look in the brain, right? Because we have different anatomical or approximately grossly anatomical areas. And as you can see, again, from theta and above, we seem to be doing a decent job. We looked at another invasive data set from humans, which was brought from another group. What this data set does is they're looking at depth electrodes, so maybe a little bit improved anatomical certainty there. And we also see there that, that we have good success with our method to actually expose this architecture in the measurement. Finally, after all, Golan did his work here and also with Ilke in rodents. So the next thing we wanted to ask is, what about other species? So what I'm going to show you now is basically, and the contrast is notoriously bad on this projector, but I hope you do see that we are basically looking at 11 rats. This is hippocampal recordings. And by and large, we're seeing quite nice agreement with kind of the method exposing this architecture in the data. It's nice kind of to compare that to the hippocampal recordings from the human, which is what I added now. And what you can see is that there is kind of, at least in part of the spectrum, there's quite nice agreement. Yeah, and there's a little note which kind of gets me excited, so maybe there's a few other geeks here in the crowd. It's not going to be a huge surprise, but it's nice to see it coming from the data. I told you we're identifying things basically in an automated fashion. And it was very easy and trivial for us to say, well, the biggest peak in the human is the alpha. And what you can see here is that next to our alpha peak of the human, we have also a very, very strong peak. And you know, this is rat hippocampus. When I read the rat hippocampus literature, often I see kind of weird stuff like, you know, from really, really good groups. Like, we took the frequency between 6 and 12 hertz, right? So I think that, you know, it's very prominent and called alpha in the human. And this kind of is in such agreement that I would argue this is probably what most rodent researchers in hippocampus would call theta, right? The theta oscillations. So I hope you enjoyed that detour. So rhythmicity uncovered a universal architecture. The electrophysiological spectrum is divisible into two categories. We have a high rhythmicity band, and we also have a novel low rhythmicity band. And you might have noticed that until this point, I didn't say much about it. I mean, maybe semantically we could process, right? If there's a high rhythmicity, we know that how it's calculated. It means that it lasts more cycles. I didn't really say much about what does it mean to be significantly below the noise ribbon, to be significantly low rhythmicity band. But there is this organization, and in order to better understand what it might mean, we are going back to the single trial manifestations of rhythmicity. And we're going to do that by doing a burst analysis of EEG data, and we're also going to look at brain stimulation data. So the burst analysis, if you will, you know, I kind of started the talk coming from one place, which is basically, oh, we look at power, we look at rhythms. Oops, we discovered this other thing. But of course, in another universe, people are looking at bursts first. And so basically, I'm kind of taking the other end to the story and saying, let's look at the bursts and understand whether bursts and the way we quantify bursts could account for the low rhythmicity dynamic. So how do we look at bursts? We basically filter our data, or quantify power in our data in different frequencies. And for each frequency, we will be kind of identifying events in which we have activity that is higher, or power that is higher than the 90th percentile. That is how we identify bursts. And what I'm showing you here is the duration of the burst quantified in cycles. Okay, so each frequency, of course, will have a different cycle length. And what you could immediately see is that the duration, or the amount of cycles we have in the green bands, let's call them, the bands in which we find high rhythmicity, is longer than the duration of the burst that we see in the low rhythmicity bands. We could also quantify the rate of bursts. And again, we can look at the bands associated with high rhythmicity and see that by and large, those bands have lower rate of bursts compared to their neighboring bands, which are low rhythmicity. So low rhythmicity is associated with a higher burst rate and a shorter burst duration. This is kind of trivial, but someone might say, okay, but Ayelet, don't you remember that lesson about not comparing between different frequency bands? Like, that could be kind of messy, right? I mean, and you couldn't even see it, right? Generally speaking, if we look at the frequencies from low to high, there is this kind of general trend where we have more rates of bursts as the frequencies are faster. So in order to do that, we basically, again, created for each frequency the phase-shifted surrogate in order to compare directly. And what we can see here, it's a lot of details, but generally speaking, if we look at the rate, for example, in the alpha band, we could clearly see that the surrogate data has more bursts than the actual data. And we see the reverse if we look at a low rhythmicity band. Okay, and you can take in as much as you want from this figure, but this is kind of a first step towards saying maybe our universal architecture is doing a pretty good job in identifying high rhythmicity states, which we've learned a lot about in this building from these people, and also doing a good job in statistically helping us discern what are bands in which we're seeing burst activity, which is also a rhythmic phenomenon, except it's short-lived. It has a higher rate and a shorter duration in terms of cycles. Another approach we could take is a disruptive approach. So, and this is work so Golan left my lab recently, moved to Cambridge, and collaborated with Meito Crespo Garcia from his postdoc lab there. She was about to start a TMS experiment, and we were able to squeeze a little proof of concept into her experiment, and she was generous enough to let us do that. So Meite was presenting pulses to a frontal site, and the site is really purely determined by her research program. I would have loved to stimulate a different site, but that is the site we had, and beggars can't be picky. And basically what we wanted to look at is we wanted to see how the architecture is responding when we show different protocols of stimulation. So we had a rhythmic protocol of stimulation. We identified individual subjects' alpha band and stimulated with 6 pulses at their individual alpha band. We then also had what we call alpha arrhythmic. So it's not exactly alpha, except that it keeps the first and the last pulse timing, and it disperses the other pulses randomly in a way that is not temporally structured. So that would be an example of taking what we call a high rhythmicity band and seeing what are the consequences of rhythmic stimulation and arrhythmic stimulation on that band. We didn't just do that for alpha. We also did that for beta 1, which is a low rhythmicity band, and for beta 2, which is a high rhythmicity band. All these defined based on our methods with each subject's individual frequencies. And finally, we also did the single pulse. So we just gave a pulse, basically a perturbation to the system, and saw, what is going on with our rhythmic architecture when we do that. And that is actually the data I will show you first. We're going back to our within trial description, the WTPL, that basically shows us, what happens from one cycle to the next when we present the pulse. These figures are prettier in reality. The contrast here is kind of killing it. But what you can see is that we have, and I'm just keeping the LaVie here, the rhythmicity scores from previous parts now, frequency is matched, so it's on the Y. But what you could see is that when we just perturb the system with a single pulse, we see the WTPL increase around those frequencies in which we had low rhythmicity. Okay? So that is one kind of perturbation. And that is kind of consistent with this idea that maybe I described it before for the rate coding. I'm reminding us no food fights today. But that is kind of consistent with an idea that maybe these short-lived dynamics are really kind of, potentially a way in which the system is kind of negotiating inputs, right? We have a very clear, strong magnetic stimulation. We see this kind of drama, and the drama is pretty much kept to these low rhythmicity bands. When we stimulate rhythmically at alpha and at beta and at beta, so beta 1 and beta 2, basically the stimulation is driving the response at the frequency we stimulate, and that would be frequency 0. So we're aligning the frequencies to 0 here. The interesting question is what will happen when we create arrhythmic stimulation? Right? And the prediction is that if this is a disruptive arrhythmic situation, it should primarily disrupt rhythmic bands, whilst the low rhythmicity bands actually might kind of jibe quite well with this type of input. And that is basically what we find. So I'm highlighting first the rhythmic bands, and I'm showing that when we create a stimulation that is rhythmic, and this is all baseline, so we see a decrease in the WTPL versus baseline. And when we provide this arrhythmic stimulation to beta 1, which is a low rhythmicity band, we actually see an increase in the WTPL. So we're looking now, basically this was all about looking at the single trial manifestations of rhythmicity. And we're entertaining the possibility that high rhythmicity is associated with kind of a more sustained dynamic, and low rhythmicity with bursts. And I guess the good news is that with this characterization of spectral phenomena, there's actually room for both types of dynamics. Not only room for both types of dynamics, but also one tool that could actually statistically describe them. The last part of my talk is going to be dedicated to understanding whether this rhythmicity architecture might uncover modes of neural dynamics. And I'm going to show kind of a few preliminary thoughts on that. I limited myself. There's a few examples, but I limited myself to a couple, which I thought are suggestive and are easy to digest at after a long talk, which is a post-lunch talk. So the first way to target this is basically to go to the bread and butter of EEG, eyes open and eyes closed. And when we go to the eyes open and eyes closed case, we see, first of all, what we all expect to see, which is that in the alpha band, when we close our eyes, there's an increase in the rhythmicity. This is now with our measures, but this is something that is very clearly seen in other measures as well. Now, with this hypothesis that the sustained activity might be related to some kind of maintenance function in the system, it is quite interesting to notice that not only the alpha is increasing its rhythmicity, but also the rhythmic beta. So the band in beta, which generally shows high rhythmicity, is also increasing its rhythmicity under eyes closed conditions. So this is a very, very kind of simple functional link, potentially linking different frequency bands, which are highly rhythmic. So maybe a suggestion that indeed, when we're kind of in this eyes closed situation, we're less sensitive to external stimulation. We are maybe more busy maintaining an internal state. The next example is a little bit more complex as things go. And this is, again, looking at Gall's data from the patients. So let's walk through this. I think it's pretty cool, and I like showing it here because I think it's the best we could do to get closer to the type of signals that many of you are looking at. So in these data, basically, in ECOG literature in general, I suppose, often people will look at the high gamma response as an approximation of spiking, okay? So what Golan did here, let's move this a little bit, there we go. So what we're looking at here is we quantify the high frequency gamma, again, an approximation of spiking. If there's spiking on this slide, I apologize. It's always only approximation. I don't have spiking for these data, but we could maybe for other data, but not today. So we approximate spiking with a broadband or the high frequency gamma response. And we're looking at all of the data, breaks included, everything. How does this high frequency correlate with the amplitude of the low frequencies, okay? And this is the general profile that we see over many different contacts or different sensors. Now, a nice thing that Golan noticed in Gall's data, and I think it's also documented in Gall's paper, is that not all units respond to the visual external stimulation. So what these subjects were doing, they had a task, they were seeing visual onsets, and some units increased firing, some contacts demonstrated increased firing in this high frequency approximation of firing rate in response to the stimulus. If we look at what this activity, or with which frequencies this activity correlated, what we notice is that this input coming from the outside world basically created a higher correlation with the bands in which we see low rhythmicity, okay? Those are not the only type of units out there or contacts out there. There were contacts that showed the opposite. So there were contacts in which a stimulus onset actually decreased their firing rate, or their approximated firing rate. What does that mean? Well, it means that that unit is probably doing something else, right? And the stimulus coming on is kind of stopping whatever it is it was doing. When we look at which frequencies in low frequencies kind of shape that response, what we see is that the high rhythmicity frequencies seem to be very correlated under these conditions. Okay? And this is a first suggestion showing that, you know, basically the units that are doing something else, they're not sensitive to the external world, they're actually silenced by the external world. Those units seem to be correlated with frequencies, which I just told you might be associated with maintaining some kind of ongoing state. And vice versa, stimuli that were sensitive to environmental stimulation, sensory stimulation, those actually seem to increase their relationship with the bands in which the rhythmicity was low. So I think this is very preliminary in terms of the mechanism that I'm proposing here, but I thought it's always nice to try and get towards a mechanism and towards an understanding of how this architecture might actually uncover modes of neural dynamics. And the kind of model that we're toying with or trying to kind of investigate is that sustained bands signify maintenance of ongoing brain states, while transient bands signify the brain's response to change, and in this case, sensory stimulation. It's almost time to wrap up, so I will wrap up. La vie, rhythmicity, is a tool to define the rhythmicity spectrum that allows us both an objective definition of bands. For many of you, particularly non-invasive physiologists here, this is big news, and we plan to make these tools available, of course, and hope to see people using them and discovering more things with them. And of course, you know, we talk about a higher frequency resolution because basically I kind of showed you today novel categories in the frequency space. I like to think about it not so much as a higher frequency resolution because that kind of has its own meaning. I like to think of it as like imagine the world in black and white and now you've added color, right? So there's this kind of another category of phenomena that we can describe now. Rhythmicity uncovers a novel universal electrophysiological architecture. High rhythmicity bands overlap with canonical bands. They signify maintenance of ongoing brain states. And the novel low rhythmicity bands are dominated by transient bursts and signify change. Rhythmicity specifies a powerful, replicable, and accessible feature set for neurotechnology and diagnosis, as well as for cross-species comparison. We've seen rats. We look forward to analyzing more data from other species. And also, I didn't have a lot of time to talk about this, but it really is a very powerful tool if we're starting to think of things like pathology, or if we want to think about aging, or just looking at specific changes within an individual subject, because we actually have good and solid tools to characterize these dynamics within a subject. Finally, there's room for both phase and rate codes, so you know, we don't need to panic. I need to thank Golan Kravat, with whom who has led this work and with whom it's been really good times moving forward. Rick Hansen and Maite from Cambridge, as well as Mike Anderson, who is Golan's current postdoc advisor, and he's letting him to do all this fun stuff on rhythms. From the Hebrew University, Gal Vishne and Leon were instrumental and insightful in letting us use their data and commenting on the manuscript, as were also Flo and Neil from the lab. I am starting a group at UCL. There is a position that will be advertised soon, so talk to me if you are interested in these topics. And just to convince you that we do time, and to remind you that the posters are still up. This is the rest of the team. Just a couple of weeks ago in my Minds and Moments conference on timing in Berlin, the posters are still out. So if you really want to hear about process-based or the neural dynamics of temporal decisions, that content is out there, and I'm very curious to receive your inputs and questions. Thanks for your attention.